Here we have a phylogenetic tree of the opisthocons. The opisthocons include the animals, fungi, and various groups of protists. Now, all of these groups are thought to have evolved from a single unicellular flagellated ancestor. Once they diverged from each other, the animals and the fungi groups are thought to have separately evolved multicellular structures to them. Now, if we take a closer look at the fungi can be divided into several different phylums. The Chytrid fungi did separate off much sooner than the other fungal species, and we're going to talk about that one first, but it'll be quite unique compared to the others. Now, the Chytridiomycosis, this is what we commonly call the chytrids. These diverged early, and so they're going to be quite a bit different. And these include both decomposers and also some parasites. Now, these groups, not only do they include parasites and decomposers, but we also have a little bit of variety in their structures. We have some that form hyphae and others that are unicellular. But the most notable thing about this group is that they do produce what you're seeing in this slide here, which is called a zoospore. What's different about these spores compared to the spores of the other fungal groups that we're going to go through is that these have flagella. Our other fungal spores will not have flagella on them. So in this picture, you can see those flagella. So that's what this group is most known for. Now, one reason that this group has gained quite a bit of popularity is that they are noted for causing significant declines in amphibian populations. They are responsible for a parasite that infects amphibians and frogs specifically, and we have seen dramatic declines in the rainforest and other places where we have lots of amphibians. And it is responsible for producing pretty high death rates, and usually this is within just a few weeks or days even of infection. The Zygomycosis is most noted for a structure called a zygosporangia. Okay, so that's where the name comes from. What you're going to find is that this group includes a lot of very fast-growing fungi, and we're going to call these molds. You can see an example of that in this picture right here. This is black bread mold that you're seeing growing here, and they can appear very quickly, and within just a day or two, we would expect that this mold would be covering this slice of bread. So here's a look at the two different types of spores that can be produced by the Zygomycosis. The ones at the top here, these are asexual spores. And this would be the most common spore type. So most of the time with Zygomycosis, as long as they have plenty of food available to them, they will be reproducing asexually. These are what we call sporangium. And these sporangia would produce asexual spores. So all of the spores that are being released from these would be clones of each other. Now down here on the bottom, this is going to be sexual reproduction. And with sexual reproduction, they form the zygosporangia. So that's where the name for this group comes from. Okay, so the black structure in the middle, this would be the zygosporangium. And the zygosporangium would form zygospores. Now in this case, since this is sexual reproduction, we're going to have two sexually compatible strains. We can call that a plus and a minus. Those are going to end up fusing with each other to form a zygote, which will then undergo meiosis to produce the zygospores. So since these zygospores will result from the process of meiosis, they are going to be genetically different from each other. So here we have a look at typical life cycle for a Zygomycosis. If we start at the bottom here with just a typical mycelium growing on some type of food substance, as long as there is plenty of nutrients available, they are going to reproduce asexually. So as they reproduce asexually, they are just going to produce these structures called sporangia, and these sporangia will produce spores through the process of mitosis. Since it is mitosis that's being used to produce the spores will all be clones of each other. Those spores will be dispersed, and once they are dispersed, if they land in a favorable location, then they can germinate to produce more clones of this particular fungi. Now, when the nutrients become limited, this is when a lot of times we have sexual reproduction take place. With the Zygomycota, in order to have sexual reproduction, we need two different strains that can mate with each other. We tend to call these a positive mating type and a negative mating type. These will secrete pheromones, which are chemicals, so that they can recognize each other. And then once they recognize each other, they will grow towards each other so that they do come together and we will get plasmogamy. Plasmogamy would be fusion of the cytoplasm. And notice that when the cytoplasm fuses, we're going to have a bunch of nuclei there in close proximity. This would be the heterokaryotic condition. So we've got multiple nuclei there, and they are different genetically from each other. They will fuse to form the zygosporangia, and the zygosporangia within that we will have karyogamy, which is the fusion of the nuclei. Once we get fusion of the nuclei, that then would be the zygote. The zygote then undergoes meiosis. Since it undergoes meiosis, it would produce spores that are going to be genetically different from each other, and these are haploid spores, and those would then be dispersed. So note that we have two different structures here. We have one for asexual reproduction, and we have one for sexual reproduction. Both spore types would be haploid spores, and both spore types would be dispersed into the environment, and as long as they land in a favorable location, then they can germinate. There are some species in this group that actually are able to aim their spores even. So here's just one example of that. These fungi can turn the sporangium so that they can really shoot those spores in the direction that seems the best and seems like it will give them the most favorable chances for germination. The Glomeromycota are very closely related to the Zygomycota. Now, these are mostly known for their role as arbuscular mycorrhizae. So they form relationships with plants. These would be symbiotic relationships. And if you'll recall from earlier in the chapter, the arbuscular mycorrhizae are the ones that insert through the cell wall. So these actually pass through the cell wall of plants, and you can see that in this picture here. So we have the fungi up here. This would be the hyphae. And down here would be our plant cells. So if your cell wall for the plant is right here, notice that you've got the fungi hyphae extending right through that, so that they are going to be inside the cytoplasm and they will be able to absorb nutrients from the cytoplasm. They provide things for the plant, and then the plant is going to provide carbohydrates back to the fungi. Now, about 90% of plants that we know of have some type of a relationship with Glomeromycota. So these are very important fungi for plant symbiotic relationships.